Welcome back to Hidden Wisdom Academy Biology. In this lecture, we'll learn about the anatomy of roots, both dicot and monocot. We'll see how their structures differ, what each tissue does, and how they look under the microscope. We'll also cover their main structure, functions, and key points. If you missed part 1, the introduction to plant anatomy, watch that before this. The link is in the description, in the cards, and in the pinned comments. Also, don't miss our daily biology podcast for quick revision. So, without further ado, let's get started. As always, in this slide you can see the syllabus and PYQ topics with their years, so you know what to expect in your exams. Now let's look at the primary structure of a dicot root. The outermost layer is the epiblema or rhizodermis, with root hairs that absorb water and minerals. Below it is the cortex, made of parenchyma cells that store food. The innermost layer of the cortex is the endodermis, with Casparian strips that control the movement of water and minerals. Inside the endodermis is the pericycle, which helps form lateral roots and cambium for secondary growth. The vascular bundles are radial, with xylem and phloem arranged alternately. The pith is small or absent. The key feature of dicot roots is secondary growth. Examples are bean, pea, and sunflower. Let's look at the primary structure of a monocot root. The outermost layer is the epiblema or rhizodermis, with root hairs for absorption. Below it is the cortex, made of many parenchyma cells with spaces for storage. The innermost layer is the endodermis, with Casparian strips that control water and mineral movement. Inside it is the pericycle, which forms lateral roots but not cambium, so no secondary growth in monocots. The vascular bundles are radial and polyarch, meaning many xylem and phloem patches arranged alternately. The pith is large and well developed. The main difference from dicots is the absence of secondary growth in monocot roots. Examples like maize, grass, or wheat. Let's quickly compare dicot and monocot roots. Dicot roots, like beans and peas, have a small or absent pith, radial vascular bundles that are tetrarch or pentarch, and a pericycle that forms cambium, allowing secondary growth. Monocot roots, like maize and wheat, have a large pith, polyarch vascular bundles with many xylem patches, and a pericycle that only makes lateral roots, so there is no secondary growth. Both types have epiblema with root hairs, a cortex with parenchyma, and an endodermis with Casparian strips. The main difference is cambium and secondary growth, present in dicots but absent in monocots. This table will help you remember these key points for exams and practicals. Let's see the functions of roots. Roots anchor the plant in soil and absorb water and minerals through root hairs. The cortex and sometimes pith store food, mainly starch. The xylem and phloem transport water, minerals, and nutrients throughout the plant. Roots also produce growth hormones like auxins that help plant growth. Some roots are modified for special purposes. Prop roots in maize and banyan give extra support. Storage roots like carrot and radish store food. Respiratory roots in marshy plants like Avicennia help in gas exchange. Aerial roots in orchids and banyan help in support and absorption. In practicals, focus on identifying epidermis, cortex, endodermis, pericycle, xylem, phloem, and pith in dicot and monocot roots under the microscope. This is an important NET topic. If you want to study root functions in detail, the link is in the pinned comment with Part 1, Introduction to Plant Anatomy. Let's focus on practical observations of roots. In dicot roots like beans and sunflower, the vascular bundles are radial, with xylem and phloem arranged alternately. The pith is small or absent. The pericycle forms lateral roots and cambium. Under the microscope, identify epiblema, cortex, endodermis, pericycle, xylem, and phloem. In monocot roots like maize and wheat, the vascular bundles are radial but polyarch, the pith is large and well developed, and the pericycle makes lateral roots only, with no cambium. Focus on the same tissues under the microscope. During practicals, carefully observe the transverse section, note the shape and arrangement of xylem, identify cortex layers and endodermis with Casparian strips, and compare Dicot and Monocot vascular arrangements. In exams they often ask about tissue identification, radial versus polyarch bundles, pith size, and cambium, so this observation is very important. Here is the diagram of Dicot and Monocot roots. In dicot roots like bean or sunflower, the outermost layer is epiblema with root hairs, followed by cortex, endodermis, pericycle, and radial vascular bundles with tetrarch or pentarch xylem and phloem. The pith is small or absent. In monocot roots like maize or wheat, the epiblema is followed by cortex, endodermis, and pericycle. The vascular bundles are radial and polyarch with many xylem patches, and the pith is large. There is no cambium, so secondary growth is absent. Let's practice few questions to test your knowledge. Let's start with question 1. Which of the following is true for monocot roots? This is a PYQ from NEET 2023. Option A. Vascular bundles are radial and tetrarch. Option B. Pith is small or absent. Option C. Pericycle produces lateral roots only. Option D. Secondary growth occurs. Explanation. In monocot roots like maize and wheat, the pericycle only forms lateral roots and does not produce cambium, so secondary growth does not happen. The vascular bundles are radial and polyarch. The pith is large and well developed, unlike in dicot roots where it is small or absent. The key point is, the pericycle in monocots forms lateral roots only. So, option C is the correct answer. Pericycle produces lateral roots only. Now, let's move to question 2. The transverse section of a plant part showed polyarch, radial, and exarch xylem, with endodermis and pericycle. The plant part is identified as. This is also a PYQ from NEET 2021. Option A. Monocot root. Option B. Dicot root. Option C. Dicot stem. Option D. Monocot stem. Explanation. This transverse section shows a monocot root. The vascular bundles are radial and polyarch, with many xylem patches arranged alternately with phloem. The xylem is exarch, meaning the protoxylem is towards the outside. The presence of endodermis and pericycle confirms it is a root. So, this is a monocot root. So, option A is the correct answer, that is monocot root. Here are some common mistakes to avoid. First, don't mix up dicot and monocot roots. Dicots have a small or no pith and can do secondary growth, while monocots have a large pith and no secondary growth. Second, check the vascular bundles. Dicots are radial, tetrarch or pentarch, monocots are radial, polyarch. Third, know the pericycle function. In dicots, it forms lateral roots and cambium, in monocots only lateral roots. Fourth, note the protoxylem position. Monocots are exarch, outside, dicots are endarch, inside. Fifth, label diagrams correctly: epiblema, cortex, endodermis, pericycle, xylem, phloem, and pith. Finally, don't confuse root tissues with stem tissues. Avoiding these mistakes will help you score better in exams. That's the end of today's lecture on root anatomy. We covered dicot and monocot roots, their structure, functions, practical observations, and key differences. Revise the diagrams and remember the main tissues. In the next lecture, we'll study stem anatomy, dicot and monocot stems, their structure, functions, practical observations, and differences. Revise roots well and get ready for stems. Don't miss our daily NEET MCQs and PYQs for daily revision. See you in the next lecture.